Good morning. Today we are going to talk about the ultrastructure and biological function of matrix vesicles in bone mineralization. So bone is a living mineralized tissue which is composed of calcium phosphate and a variety of organic materials, with collagen being the most abundant. So in bone there are primary mineralization and secondary mineralization stages. In primary mineralization there are two phases where the matrix vesicles mediate the mineralization first, and then the collagen mineralization happens. In the secondary mineralization, the physicochemical deposition of calcium and phosphate ions, which is presumably transported by the osteocytic network inside the mineralized bone matrix. So first the matrix vesicle form a mineralization, then comes the collagen mineralization. Once the mineralized bone matrix is formed, the extracellular calcium and phosphate ions are drawn inside. That is called the secondary mineralization. So during the process of matrix vesicle mediated mineralization, the osteoblasts regulate the vesicle synthesis as well as the activity of the membrane transporters and enzymes with which the matrix vesicles are equipped. So the osteoblasts produce the matrix vesicles, and they have the matrix vesicles have their membrane transporters and enzymes. So these are the osteoblasts, and they secrete the matrix vesicles. These matrix vesicles initiate the mineralization, and then they come out and into the collagen matrix, and then they form an osteoid, and then the osteoid gets further mineralized to form the mineralized bone. Now we talk about generation of calcium phosphate crystals in the matrix vesicles. Nucleation of calcium phosphates in matrix vesicles. They are matrix vesicles are nothing but, we already discussed, they are small extracellular vesicles enveloped by plasma membranes, which are secreted by the osteoblast into incompletely mineralized osteoid. So this is how I will show the picture later. The matrix vesicles, these are the matrix vesicles, which are the 10 images of the matrix vesicles. An influx of calcium and phosphate ions through their membrane transporters and enzymes initiate calcium phosphate nucleation and subsequent crystal growth inside the matrix vesicles. So calcium and phosphate ions enter the matrix vesicles through the membrane transporters and enzymes. The plasma membranes of matrix vesicles are rich in acidic phospholipids such as phosphatidylserine and phosphatidyinositol, which have a high affinity for calcium because of phosphate residues to produce a stable calcium phosphate phospholipid complex. So they have a high affinity for calcium. So this is how a matrix vesicle attracts all these calcium and phosphate ions and starts the nucleation. And how does the crystal growth in matrix vesicle happen? Initially, the calcium phosphate is in the amorphous form, which appear inside the matrix vesicles, and then they are converted into the crystalline calcium phosphate, that is hydroxyapatite. They form needle-shaped or ribbon-like crystalline calcium phosphate from a stellate assembly growing in all directions, and then come out of the matrix vesicle, penetrating the plasma membrane to form calcified nodules. So once they form that star-like thing, they form the calcium nodules, and this is the TEM image of a calcium nodule. We have the osteoblasts here, and the matrix vesicles have come, and they have deposited the calcium nodules are being formed. Distribution of calcium phosphorus surrounding matrix vesicles. And how does this mineralization happens around the matrix vesicle? Calcium is abundant in the close proximity of the matrix vesicle or calcifying nodules, while phosphate is conjugated with organic substances in somewhat distant from this matrix vesicles. So in non-mineralized areas of osteoid, the extracellular meshwork of organic substances may limit the production of hydroxyapatite and inhibit the precipitation of mineralized crystals by controlling the spatial distribution of calcium and phosphate, even if the extracellular fluid is subsaturated with those ions. What are the biological functions of the enzymes and membranes transporters of matrix vesicle mediated mineralization? So the biological actions of membrane transporters and enzymes are to synthesize the phosphate monomer rather than calcium, and to enable the influx of phosphate into the matrix vesicles. So we have seen that calcium is abundant inside the matrix vesicles, but phosphate has to be drawn in. These biological actions only produce the phosphate monomers, which is essential for the precipitation of the calcium phosphate. So there are few enzymes, namely the ectonucleotide pyrophosphatase phosphodiesterase, which is called ENPP1 for pyrophosphate synthesis. So the ENPP is a membrane of the ENPP family of proteins which possesses, so this is the structure of the ENPP protein, which has a catalytic domain and a nucleus-like domain, and it has a somatomeric V-like domains. This is the somatomeric V-like domains, catalytic domain and the nucleus-like domain. So the catalytic and the nucleus-like domains are for bone mineralization, whereas the SMB domain is for insulin signaling. The catalytic activity of ENPP located on the cell membranes of osteoblasts generates inorganic pyrophosphates. So this PPI, we know already, it is an inhibitor of mineralization. It inhibits the mineralization by binding to nascent hydroxyapatite crystals and thereby preventing overgrowth of these crystals. So in human infants, severe ENPP1 deficiency was shown to be linked to a syndrome of spontaneous infantile arterial and periarticular mineralization. What happens when there is ENPP1 deficiency? There is less PPI. PPI is an inhibitor of mineralization. When PPI is less, there is many pathological mineralization happening. So this ENPP deficiency causes pathological mineralizations. The next is ankylosis. This is a multiple pass transmembrane channel. This is a channel. ENPP is an enzyme. So ANK is encoded by mouse progressive ankylosis gene and appears to function as a multiple pass transmembrane channel, allowing anions and PPI to pass through the plasma membrane from the cytoplasm to the outside of the cell. So what happens? This ANK membrane protein helps in transporting the PPI anions from inside the cell to the extracellular area. So ANK mediated regulation of extracellular PPI levels is a possible mechanism which controls the tissue mineralization and susceptibility to arthritis. To avoid pathological mineralization, many cells may transport intracellular PPI into extracellular fluid by mediating ANK. So infants carrying ANK mutation exhibited 3 to 5-fold decrease in extracellular PPI in contrast to the stimulatory effect of intracellular PPI. The next is TSNL-AP, which is we have already seen, tissue non-specific alkaline phosphatase. So which is an important enzyme that initiates mineralization in bone, which is a 86 kD homodimeric protein enzyme belonging to the glycosphosphatidyl anchor enzyme associated with the cell membrane. So TNSLP is not uniformly localized on the cell membranes of mature osteoblasts, which show distinct cell polarity. So in the region of osteoids, it is more expressed, but not throughout the cell membrane. So TSNLP hydrolyzes the PPI into phosphate monomers. This is how from PPI, which is an inhibitor, the phosphate monomers are formed to promote mineralization. It is a potent inducer of mineralization, and the prevention of crystal growth is blocked, which means mineralization is stimulated, and the phosphate monomers are transported into the matrix vesicles by the PIT1, which is the co-transporter of sodium and phosphate. And the last is the phosphohydrolase activity of phospho-1. Phospho-1 is also is in the matrix vesicles. What is phospho-1? It is highly expressed in the mineralizing cells like bone and cartilage, and is specifically responsible for the increment of local ratio of phosphate or PPI inside the matrix vesicle for initiating crystallization. So TSNL-ALP and the phospho-1 do not compensate for their function in crystallization. How? TNS-ALP is anchored on the outer plasma membrane of the matrix vesicle, but does not exist inside the vesicle. So the phosphate monomer generated by this alkaline phosphatase must be transported to the matrix vesicles, presumably by means of sodium phosphate, that picotransmitter PIT1. While the phospho-1 is thought to function inside the matrix vesicle to catalyze the phosphocholine, a constituent of plasma membrane, dividing it into choline and phosphatase. So if we see the picture, it will be clear for us to understand. So this is osteoblast. The osteoblast is here, and the osteocytes are inside the osteoid, which is getting deposited, the osteoblast. So the TNSLP is the organelle which is present on the outer membrane of the pre-osteoblast and the osteoblast. And the ENPP, which is present here, which comes to the extracellular space and forms the PPI. PPI is the inhibitor of mineralization, which means it inhibits the matrix vesicle. This PPI is activated or converted into phosphate monomers by the alkaline phosphatase, which helps in the mineralization. This phosphate enters through the pit 1 channel, and the calcium transporter transfers the calcium. So calcium and phosphate are saturated here and they form the crystal here. Now the crystals go out. For this, the plasma membrane of the matrix recycle helps, where the phospho 1 converts the phosphocholine into phosphate and choline. So this phospho 1 acts here, and we see the TSNL. This is the matrix recycle where we have all the membrane transporters and the enzymes playing an important role in the mineralization here. So we see the Golgi apparatus produce PPA, the ANK, the ankylosis cycle, that pushes the PPA out to the extracellular membrane. Then it is there present which inhibits mineralization. This is converted to by the alkaline phosphatase into the phosphates, which induces the mineralization. So for when we see the biological enzymes and transporters, we have ENPP1, ankylose, alkaline phosphatase, and phospho1 playing an important role in matrix recycle mineralization. And what is the biological function of non-collagenous proteins in the calcifying nodules? We know what a calcifying nodule is. It is a globular assembly of radially arranged needle-shaped minerals, crystals, and the calcium nodules originate from calcium phosphate crystals found in the matrix vesicles. These crystals grow in the vesicles and are exposed to the extracellular environment. The growth of the calcifying nodules appear to be regulated by extracellular organic materials such as osteopontin, osteocalcin, and matrix GLA protein, which are rich in the osteoid. So these extracellular non-collagenous substances help in regulating the calcifying nodules. The osteopontin is shown to be localized in the periphery of the calcium nodules, and therefore it might act as an inhibitor of abnormal excessive mineralization. Whereas osteocalcin and matrix GLA protein are known for containing gamma carboxyglutamic acid, which could be gamma carboxylated. That is, osteocalcin becomes gamma carboxylated to obtain a high ability to bind to calcium phosphate crystals in the bone. So, osteocalcin plays an important role in the globular assembly of the needle-shaped mineral crystals. So, when we see the images here of osteopontin and osteocalcin, we can see this golden nanoparticle loaded osteocalcin, which is found along the periphery of the calcifying nodules. And we can see how they are arranged in the cartilage and bone matrix in the intermediate zone. So this is how the calcifying nodules are formed, where the osteocalcin is gamma carboxylated, which has a high affinity for crystalline calcium, and these calcium nodules will deposit the minerals in the collagen fiber. This is the regular arrangement of the minerals in the collagen fiber, whereas when there is non-carboxylated osteocalcin, the crystals are not in a properly arranged form and they are scattered, so the mineralization of the collagen fiber doesn't happen. So this is where the gamma carboxylation of the osteocalcin plays an important role. This is where the osteocalcin is seen in a TEM membrane, which is gold nanoparticle loaded osteocalcin. When there is a warfarin is an inhibitor of gamma carboxylation of osteocalcin. So there is less osteocalcin seen here, and the mineralization is not in a proper arranged way. Here is a regular pattern of calcium nodule formation. So this talks about the non-collagenous proteins like osteopontin and osteocalcin in the mineralization of the matrix vesicles. And what about the collagen mineralization from the contact site with the calcium nodule? So the collagen mineralization begins at the point of contact with the calcifying nodules, and the theories explaining collagen mineralization are whole zone theory. So during the mineralizing phase, the gaps within the collagen fibers are occupied by small proteoglycans such as decorin and biglycan, and after elimination of these proteoglycans, extracellular calcium and phosphate fill in the gap to generate calcium phosphate nucleides and subsequently mineralize the collagen fibers from the inside. So mineralization takes place along the superhelix of collagen fibers, which are arranged in parallel but shifted at certain levels. And we always talked about collagen as a triple helix structure. So in that helix, the mineralization happens here. And this shows the helical structure of the collagen fibers, where the matrix vesicles, they form the mineralization, and from the matrix vesicle it comes out and it forms gets arranged along with the collagen fiber to form a collagen mineralization structure. And to conclude,